Hello everyone, today we will discuss about the metabolism of HDL. HDL is a lipoprotein that is synthesized in liver and in intestinal mucosal cells. The major function of HDL is to transport cholesterol from peripheral tissues to liver. So, HDL transport cholesterol from peripheral tissues to liver for excretion through the bile either as cholesterol or after conversion to bile acids. This process is known as reverse transport of cholesterol. Reverse transport of cholesterol or reverse cholesterol transport. So, let us see the steps in the metabolism. So, HDL is synthesized in liver and in intestinal mucosal cells. In liver and in intestinal mucosal cells. This is nascent HDL. Nascent HDL. This nascent HDL is composed of a bilayer of phospholipid and a free cholesterol. It is discoidal in shape. It is discoidal in shape. And it is composed of a bilayer of phospholipid and cholesterol. And it is known as nascent discoidal HDL. Nascent discoidal HDL. And it contains ApoA1, then ApoC2 and ApoE. So, a bilayer of phospholipid, cholesterol, ApoA1, ApoC2 and ApoE. This nascent hepatic HDL, nascent hepatic HDL contains ApoA and ApoC. Then nascent intestinal HDL contains ApoA. When it circulates, it acquires ApoC and E. Then this nascent discoidal HDL, it is secreted into the plasma. It is secreted into the plasma. Then ABCA1, that is ABCA1, ATP binding cassette transporter, A1. It is a HDL receptor protein that transfer cholesterol from peripheral tissues to pre-beta HDL or this ApoA1. So, this receptor, this HDL receptor that is known as ABCA1, that is ATP binding cassette transporter A1. It is a HDL receptor protein that transfer cholesterol from peripheral tissues. To mainly pre-beta HDL or to ApoA1. Pre-beta HDL means this is ABCA1. This pre-beta HDL means this ApoA1 will form pre-beta HDL when in association with little amount of this phospholipid and cholesterol. So, after associating with a minimum amount of phospholipid and cholesterol, this ApoA1 form pre-beta HDL. This pre-beta HDL is the most potent form of HDL that will induce cholesterol efflux from tissues. That is pre-beta HDL. This pre-beta HDL is the most potent form of HDL that will induce cholesterol efflux from tissues. So this ABCA1 receptor, that is ATP binding cassette transporter A1, it is a HDL receptor that transfer cholesterol from peripheral tissues to pre-beta HDL or ApoA1 or to this nascent discoidal HDL. This time LCAT, that is lecithin cholesterol acyl transferase, LCAT, lecithin cholesterol acyl transferase of plasma binds with this nascent discoidal HDL, and this enzyme is activated by this ApoA1 and ApoC2. The main function of this LCAT is that it transfers acyl group from this HDL phospholipid, that is lecithin, to cholesterol. Lecithin to cholesterol, and it forms lysolecithin and cholesterol esters. That is the function of LCAT. The function is that it transfers acyl groups from HDL phospholipid, that is lecithin, to cholesterol, to free cholesterol, and it forms lysolecithin and cholesterol esters. Then this lysolecithin, it is released from this HDL to plasma, where they will bind to albumin. It will release to plasma from this HDL and it will bind to albumin in plasma. Then what happens to this cholesterol ester is that these cholesterol esters are transferred to the this central core of HDL particle. It will transfer into the central core of HDL particle. So by this discoidal HDL is converted to spherical HDL, and it is known as HDL 3. So it will contain cholesterol esters, ApoA1, C2 and ApoE. So nascent discoidal HDL, it is converted to spherical HDL, that is HDL 3. Then from HDL 3, cholesterol ester will move to other lipoproteins such as this VLDL, chylomicrons, etc. So from HDL 3, cholesterol esters will transfer to VLDL or chylomicrons in exchange of triacyl glycerol. This is by CETP, that is cholesterol ester transfer protein. So by this time, this HDL 3 is converted to HDL 2. It is converted to HDL 2. That is, it will become bigger than that of HDL 3, and it will become CE triacyl glycerol, then this A1, C2 and E, and it is converted to HDL 2. This HDL 2 is larger in size and less dense than that of HDL 3. So it is bigger than that of HDL 3. Then in liver there is receptors, that is known as class B scavenger receptor B1. Class B scavenger receptors, scavenger receptor B1, that is SRB1. These are HDL receptors. These HDL receptors will bind with this SRB1 here, HDL receptor in hepatocytes. These HDL receptors will bind with the HDL2 through ApoA1. It will bind to HDL2 through this ApoA1. By this cholesterol ester enters the liver. This is for excretion through bile, either as cholesterol or after conversion to bile acids. This process is known as reverse transport of cholesterol or reverse cholesterol transport. So class B scavenger receptor B1, that is SRB1, that is HDL receptors will bind with HDL2 through ApoA1. By this cholesterol esters enters the liver for excretion through the bile either as cholesterol or after conversion to bile acid. This is known as reverse transport of cholesterol. Then from HDL2, this HDL2 to HDL3 is possible. So HDL3 is reformed either after delivery of this cholesterol esters to the liver through this SRB1 or by the hydrolysis of this triacylglycerol or triacylglycerol and phospholipid, HDL2 phospholipid by hepatic lipase. So by this HDL2 is converted to HDL3 or this HDL3 is reformed. This interchange of HDL2 and HDL3, it is known as HDL cycle. HDL cycle. So this HDL3 is reformed either after delivery of this cholesterol esters to the liver through this SRB1 or by the hydrolysis of HDL2 phospholipid and triacylglycerol by the enzyme hepatic lipase. This interchange of HDL2 and HDL3, it is known as HDL cycle. So this is about HDL metabolism. This is today's topic. Thank you for watching.